Good morning, everybody. Welcome to the webinar. We're going to start at 8:03 and give people a chance to trickle into the webinar. Good morning, everybody. Welcome to the webinar. We're going to get started in a minute. Hi, everybody. Welcome to the RCN NSF funded work webinar on SARS-CoV-2 and wastewater. It's nice to see everybody this morning. This is a webinar for what we're calling junior scientists who have been working so hard over the last year to inform the nation's and our university's responses to the pandemic. Many of them have switched their research trajectories and put their PhD research, their main research on the side to contribute to this research area. So we're very grateful for that. And this webinar is a celebration of their hard work and accomplishments. Today we're happy to have two great speakers. We have Ayella Maile-Moskowitz from Virginia Tech. And then we have Yi Li from University of Tennessee. Ayella is going to go first. And just to remind you all how this works, we are taking questions in the Q&A. As the webinar presentation wraps up near 20 minutes, please put your questions in the Q&A, not in the chat. And if you are looking at the Q&A and you see a question that you like, please upvote it. And then Jill and I will be the moderators to ask questions of the speakers. So with that, we're going to go ahead and get started. Ayella, you can go ahead and share your slides. Looks good.
Okay, good morning. My name is Ayella Maile-Moskowitz. I am a PhD student at Virginia Tech, and I'm going to be talking about our wastewater surveillance of SARS-CoV-2. So the goal of this research was really to inform the university where they should be kind of focusing their clinical testing. So probably similar to what many other universities are doing. We have a bunch of different clinical testing on campus. We have symptomatic testing with contact tracing. We have asymptomatic testing, and then we also have random mandatory testing. And this is really where the wastewater side comes in for us. So the university will each week tell students they've been selected to be randomly tested. And so when we have a positive result from a dorm or dorms, they can take that information and then oversample a certain dorm or dorm. So it's not truly random. And we also have an isolation dorm or dorms on campus that we are also sampling. So just to talk about our campus layout, so the figure on the left is a breakdown of the buildings. And so Virginia Tech's campus is split into an academic side and a residential side. And so on the one hand, we kind of have the academic side up at the top, and the residential is in blue. And so on the right, we have a layout of our sewer system. And similar to kind of how that is laid out, the water flows from the academic side and the residential side to this central line. And then that line flows out of campus. It combines with the town flow and goes to the wastewater treatment plant. So based on this sewer layout and just logistics of how many samples we could take at once, these are the sampling locations that we came up with. Some of the locations are one dorm, but a lot of them are 2, 3, or more dorms in one sample. We also have one sample that represents kind of most of campus, which is our location B. Our location C is a research facilities location, so it doesn't get any dorm flow, and it's primarily just research buildings. And then we also have our sports complex building, so that is mostly where athletes are practicing. We're getting flow from that building as well. So for this presentation, I've kind of narrowed down the number of buildings that I'm talking about. So as I mentioned, we have kind of our all location or all campus location. We also have location A, which is a little bit further downstream than most of our other sampling locations, and it captures a bunch of buildings. We have 2 locations that are 2 or 3 dorms, and then we have 2 locations that are each 1 dorm. So now to talk about our sampling methods. So when we first started off sampling, we were just collecting grab samples. I have my lovely advisors here on the left. I got to see them in the field, which was very fun. And so we've been working closely with the facilities on campus. They've been helping us with sampling, and they came up with this essentially bottle on a really long pole. And that's important because some of our sewers are 15, 20 feet deep. So in order to not have to go down into the sewer, we can just dip this bottle down into the sewer to collect samples. So that's how we started off sampling, just collecting grab samples. And then in October, I believe, we got some composite samplers that we were able to set up around campus. And for the most part, they are above ground, and they're kind of, they have this construction fence around them, though a few of them are suspended in the manholes. And the way that we kind of set things up, the tubing from the samplers, we've got it connected to some pipe, just to be able to maneuver a little bit better in the sewer and to position it, the tubing. And we have these weighted strainers at the end of the tubing that lays down in the sewer. And being so far upstream, most of the time we have low flow strainers just because the flow does get very low and is quite variable so far upstream. And so these samples, locations that we have on campus, we are sampling twice a week. And we also sample our local wastewater treatment plant every week. In terms of the lab, we are filter concentrating with electronegative filtration, and we're doing 150 milliliters after adding magnesium chloride and adjusting the pH. And after we filtered, we actually manually rip up our filters with some sterilized tweezers to help with the extraction process downstream. We then store our sample or our filters at -80 degrees until we can get them to a lab that does the reverse transcription qPCR for us. So a little bit more on that. The lab that is doing this for us, they are also analyzing the clinical samples in our area. And so when we first started to look into doing this back in August last year, we wanted to just test if their assay works. So they developed their own FDA-approved assay where they're looking at the N2, S6, and E1 genes, as well as a human-associated gene. And so our initial analysis, we spiked some heat-inactivated virus into some wastewater that we had collected, and we did a 24-hour experiment where we looked at if we could see any degradation over those 24-hour period with our samples stored at 20 degrees. We also added RNA stabilizer to some of our samples because we wanted to see if we had to do something like that, like they do for the clinical samples, if we had to do that for our wastewater samples. And we found that the assay was able to detect the virus both with and without the stabilizer. So we kind of got our green light that we could go ahead with this method. So briefly on how we get back our results, because this may be a little bit different than if you're doing the samples yourself. So we sample, or we give them one replicate per sampling location, and they then run those in technical duplicates. So we know if one or both of those technical replicates came back positive, but we only know the gene copies if 2 or more samples came back positive, which I'll touch a little bit on later. And we only have the gene copy numbers for the N2 gene because that's the only gene that they are doing a standard curve for. We are also spiking in bovine coronavirus, but their lab doesn't look for that. So we are working on getting back some RNA samples so that we can back calculate recovery based on the RNA that we spiked in, or the virus that we spiked in. And so besides just looking for COVID-19, SARS-CoV-2, we also wanted to look for some water quality parameters. So we decided to analyze our samples for ammonia, chemical oxygen demand, and total and volatile suspended solids. And the initial thought was if these data can correlate in any way to the population on a building scale, because this has been done on a wastewater treatment plant scale to be able to back calculate the population in a catchment area. But before we kind of look at all of our data, I wanted to talk a little bit more about some of the challenges that we've encountered and how we've dealt with some of these. A lot of these, in retrospect, are kind of intuitive, but for us doing this for the first time, they were definitely things that we learned. And because I spend a lot of my time looking down manholes, that's kind of what I'm going to be talking about. It turns out that if you put anything into the sewer, anything and everything will stick to that something in the sewer. And it turns out that people flush all kinds of things down the sewer that they're not supposed to. So as anybody who has worked with this can probably relate, there's all kinds of clogging and things sticking to your samplers in the sewer. The two figures on the right are actually the same manhole. So on the left, you can see the flow is flowing nicely. We have this little trough. And then on the right, things got backed up and it essentially flooded the sewer. So far, we haven't had any complaints either on the building scale or downstream in the sewer that there were any problems with this. Whenever we pull up the strainer, the flow continues on. So it doesn't seem to be impacting anything within the sewer system. But I do think that it impacts our samples. So as you can imagine, if the flow is not flowing onwards, everything is being backed up and the solids are settling out. And because our strainer is sitting in the base of this trough, we're collecting more kind of solid material in our samples. And then some of our sampling locations, the figure on the bottom left, we have a pipe will come out at the top of the sewer, but the flow is actually at the bottom. And there it combines with other buildings that we don't necessarily want to capture. So for those locations, we just continued to take grab samples. So our other major challenge was just sampling in the winter and dealing with frozen samples. We had a couple of ice storms this year as well, which was fun. And so what we did was we wrapped our samplers in isolation. We also put some pipe insulation around our tubing. And we found that when we went to set up our samplers to program them to run, we took some boiled water that we would pour over the tubing so that we could crack out the ice that had frozen in the tubing. And the only other thing that we found that worked really well was just that when there was more volume collected in the base of our sampler, it was less likely to freeze, as you could expect. And this is also when we started to run into some battery issues. So the batteries just did not like being that cold. And so even though we would set things up to run for 24 hours, it didn't always do a 24-hour period. So now that I've kind of touched on why some of the samples may have some variability in terms of like on-the-ground sampling, sometimes the grab samples especially just have a lot of extra stuff in them. So this figure on the right is just one of our more extreme examples of loss of toilet paper being caught in a grab sample. Interestingly enough, our research facility location, so that doesn't get any dorm flow, it almost always looks clear like tap water, which is the only time I've ever seen that in a sewer. And so we're still figuring out what some of the variability may be from. So is it because of the building that the flow is a little bit more variable, or is it because of the configuration in the sewer? So finally, to talk about some data. So for our water quality data, on the left is showing total suspended solids, the top right is ammonia, and the bottom right is chemical oxygen demand. And so when I first put these figures together, I thought that maybe the fewer number of people in a sample, it represents more variability. So the two box plots on the right within each figure, each have about 450 students and are just one dorm. And sorry, one more, the smallest box plot, or the lowest value, that is the wastewater treatment plant. So all of these values further upstream in the sewer are higher than at the wastewater treatment plant scale, which you could also expect. So anyway, I thought that maybe there was more variability or greater values with the fewer number of students in a building. So I went back and graphed all of the locations. And in a nutshell, it doesn't correlate. So the yellow box plots have fewer number of students. We have a minimum of 80. And then the more purple, the more students are captured in a location. So a maximum of 2,700 students. And then the gray box plots, I don't have any population data for that. So really all that I plan to show here is just that there's a lot of variability, and we don't have a super clear reason as to why some locations might have more or greater or lower values. We also looked at some correlation, and the best correlation is between ammonia and chemical oxygen demand, but it's not great. It's just an R-squared of 0.5. So finally, to talk about some of our positive samples. So we have some pretty stringent reporting. So as I mentioned earlier, the university was using our data to then oversample certain dorms. So they weren't really interested in ambiguous samples. They didn't really know what that meant and what the action should be from that. So we had some more stringent reporting where we only called something positive if 2 or more genes came back positive from our which is the same as clinical testing. They have those same requirements. So in the fall, we had about 8 samples that were positive, with 18 being more ambiguous. So here, if it's ambiguous, I'm saying ambiguous if 1 gene came back positive, or if we had multiple technical replicates come back positive. This was a shorter semester, so we didn't get our composite samplers until October, and then the students went back home after Thanksgiving and didn't come back. So it was a little bit of a shorter sampling period. And then in the spring semester, so far, we've had 11 positive with, I think, over 20 ambiguous samples. And we're currently in campus, in the community, have some quite low cases. So we haven't had any positive samples in our wastewater for the past month and a half. And we currently only have 11 students in our isolation dorm on campus, with just 14 students testing positive in the past week. So we have low number of positive students on campus, and that correlates with the wastewater that we've been seeing. So lastly, these are some very, very preliminary results. So we've also been working on doing some nanopore sequencing to hopefully be able to detect variants in our wastewater. So we're following this protocol from Josh Quick, where we're using Arctic primers to sequence or to amplify multiple different regions along the genome, and then that amplified DNA is then sequenced. We're using nanopore sequencing, and so the figure on the left, on the x-axis, is showing the position along the genome, and the y-axis is sequencing depth. So the higher that peak is, the more genes, or the more amplification we got from that certain gene region. This was one of our promising results, but most of the results didn't have great amplification or sequencing results, just because we're still working through making sure that our amplification process worked well so that we can get good sequencing, as well as just dealing with inhibition and things like that. And so we're also waiting on some back-ordered flow cells, so this is very, very preliminary. So in the future, as I've mentioned, we're continuing to work on our nanopore sequencing. We are also getting back our RNA samples so that we can look at bovine coronavirus recovery, as well as other viruses like the pepper mild bottle virus. We are also working with some other professors, Dr. Cohen at Virginia Tech, to model, swipe card data in the dorms with the wastewater data that we have, as well as the positives. And we're also going to go back and look at some of these ambiguous samples to see how they correlate with the number of students that were actually infected in a building, or perhaps it was students that came back from isolation and went back to the dorm, so that we can have a better idea of what these samples mean perhaps for next year. We haven't heard yet what the university, if they would like to continue this sampling or not, so stay tuned. And with that, I thank you for listening, and I'm happy to take questions.
Thank you so much, Ayella, for that great, clear talk and wonderful photos. So just a reminder to the audience to please put questions in the Q&A and upvote questions that you'd like to hear the answer to. So we have a bunch of questions in the Q&A, and I have some questions too. Since I'm asking questions, I'm going to ask the first one, and that's, I wonder if you could let us know what sort of ethics you had to consider in implementing this project, and whether you had to apply for IRB, and how and if you engaged with the students, or how the university engaged with the students to do the sampling, or if anyone felt, students felt violated that their sewage was being sampled, or was that just a non-issue? So I guess the two parts of it are, did you have to get an IRB? And also, did you have any other ethical issues or actions that you took in implementing this?
We did not have to get an IRB for the wastewater side of things, because we can't go back and figure out what students, contributed to our wastewater sample. As for how the students felt, the only interactions that I've had, they will ask us when we're setting up the samplers what we're doing. We had, news articles and things like that, so people knew what was going on. They weren't surprised when we told them we were sampling for COVID. They were a little disgusted that we were sampling wastewater, but I never got the impression that they felt violated. I think they thought it was kind of cool. I hope.
Okay, good. Okay, so I'll just sort of go through these questions. Do you know why you haven't been able to pick up positive samples in the wastewater if 14 students were infected based on clinical tests? I don't know if does that make sense to you?
Yes. So I would say we don't have a clear reason why. It could be that those students aren't shedding. The isolation dorm happens to be one of those locations where we have to get grab samples because the flow comes out above and then combines with other buildings. So it could be that we just missed it since it's a grab sample and not a composite. And it could be that we have other inhibition or things like that in the qPCR that we just haven't sorted out to improve our detection.
On the issue of detection, there's a question about the methods that the pre-analytical method that you chose, the filtration with the magnesium chloride, and a question as to why you chose that method as opposed to a different concentration method.
So we had the supplies. We do this similar kind of filtration for DNA. And so it was a method that we felt comfortable implementing in our lab, and we had the supplies and things like that. And then by the time we got established, we didn't want to switch and try a different method in the middle, so that this way we can compare all of our samples across our sampling period.
Okay, that makes sense. I have a question here about the sequencing data that you presented. You presented the one plot that shows the coverage or the depth of sequencing across the length of the SARS-CoV-2 genome. And one of the attendees noticed that there was a high peak near the 5' end of the genome and was wondering if you knew why there was greater depth.
The short answer is no. The greater depth, I would assume, is just that there were like more of those genes amplified, but exactly why that happened in that region, I don't know for sure. I would hope that it just means that there's more of that gene in our sample, but if that is accurate, I don't know.
Okay. I have a question here about data sharing. So, and sorry if you presented this, but in the Virginia Tech dashboard, do they incorporate the wastewater data onto that dashboard right now?
They do not. Yeah, that's just the clinical data is only there. So our information is shared internally. I think we have time for 2 more questions, and there's a bunch. So after this is over, you can take a look at those just to see how interested people were. I have one here about how challenging was it to identify your sampling locations on campus, and how sure were you that your sampling location actually contained the wastewater you thought it contained? And the person comments that on their campus, they discovered things were not set up the way they thought they were when they actually like dug in to figure out like where wastewater was coming from to different sampling locations. Did you run into anything like that? Nothing major. So we initially just took that map that I showed earlier, and we went around campus and lifted up manholes, and that was part of the process of figuring out where to sample. But we work really closely with the facilities on campus, and they are in and out of the sewers all the time. So I was relying on their expertise, that they know what's going on in the sewers.
Okay, and I'm going to ask one more question here, and that's, it's really great that you were able to work with the clinical lab in partnership to do the molecular assays, because they're doing it all the time, high throughput. So that's like a huge like a good thing about working with the clinical lab. But were there any cons? Like, what were the challenges that you've faced in interfacing closely with a clinical lab to process environmental samples?
Yeah, there are definitely challenges. I think in part we just don't have control over how our samples are processed. And so, for example, they have so many other samples because they're processing all these clinical samples. So they don't necessarily have time to kind of play with the wastewater samples and figure out what might be, you know, maybe we have more inhibition and we're just not dealing with it in certain samples. So I would say probably not having that kind of finesse to change things around. But it is really great to be able to process all of our samples at once, and there aren't any issues with the number of samples we can process.
Great, thank you. Thank you so much, Ayella, for your talk. We're ready to start the next speaker, which is Yi Li. So Yi, can you go ahead and share your screen?
Yes. Sorry, wait a minute. We'll start from the beginning. Thank you. So can you see my slides and hear me?
Yes.
Okay, cool. So everyone, hello everyone, and thanks for joining us for today's seminar. Thanks for the introduction. As she said, my name is Ayella, and I'm a PhD in the University of Tennessee. Our group is based in Knoxville, Tennessee. I'm going to talk a little bit about our work of raw wastewater surveillance for SARS-CoV-2 over 26 weeks in academic year in the University of Tennessee student residential buildings. The purpose of this project is to determine which building at the campus may have SARS-CoV-2 signals in the wastewater. This is a simple presence and absence test so that we can determine if further testing is needed. First, I want to introduce the University of Tennessee. It is a public land-grant research university. It has 11 colleagues and enrolled more than 13,000 students in fall 2020. It is a large urban university with more than 7,000 students living in on-campus dormitories, fraternities, and sororities. This is the information I get from the University of Tennessee. We can see the COVID cases trend from this figure. From this figure, you can notice that after the mid of September, the COVID cases decreased dramatically, and from the beginning of October, the number of active cases is no more than 100. This is the weekly wastewater monitoring map. For us, this campus is a residential campus with 14 residence halls and more than 3,000 rooms. In our campus, all freshmen are required to live on campus. This project, we are allowed to University of Tennessee to identify quickly buildings that may have COVID cases and help control the spread of COVID cases in campus. Sampling was conducted at the building levels once a week. The first sample were collected on September 4th, 2020, and continue to present day. This time fragments represents a significant portion of the 2024 semester, during which time the campus were closed and classes were moved online. For most of the buildings, we collected a single 500 ml sample per week. For the buildings with the highest populations, we performed a 24-hour samplers several times. From this map, you can see here is the sororities, and we have 14 sampling points. And here, the yellow represents the fraternities, and it is 15. The orange represents the dorms, the big dorms. And this is the picture that's how we take samples in the buildings. And we're taking the samples from the main hole. And also this is the way we put the 24-hour sampler into the main hole. The analyze process for each sample began with a 2-hour pasteurization at 60 degrees, and followed by a centrifugation and filtration. We use the M-counts to concentrate the RNA, and we use the K-agent kit to extract the RNA. The presence of SARS-CoV-2 was determined using the qPCR assay developed by the CDC, and we use N1 and N3 as our targets to measuring SARS-CoV-2. Also, we have detected the paper virus to maintain that we were sampling from the human sewage. This table can confirm that our method is reliable. And also we get the risk level from Arizona University. This is the standards that we use to report our results to the University of Tennessee. You can see when the concentration of SARS-CoV-2 copy numbers per liter is above 100,000, it will be a problem for that building. And from this is a big figure that contains a lot of data. And this figure shows the SARS-CoV-2 concentration variation in the time series of 47 sampling locations. To date, we have collected and analyzed 1,375 samples, with around 34% of those being reported as a positive detection. And the detection average concentration about the SARS-CoV-2 is around 1,000 viral copies per liter. And from this figure, you can also notice that the highest detection is above 100,000 virtual copies per liter. This is also a big graph from our results, which shows the weekly average concentration of SARS-CoV-2 in the dorms and the fraternities and the sororities. This is the active cases and self-isolation. The column represents the average concentration of SARS-CoV-2 in different kinds of buildings. And you can see that the blue line represents the active cases during that time in our campus. It seems like there is a relationship between them. Also, to see the data in detail, we found that the variation of the heat percentage among different dorms is wide. Unfortunately, we found that there is no significant relationship between the student numbers and the heat percentage. Also, we found that during the fall semester and the spring semester, the students' number changed in different dorms. We found the same results with the fraternities and the sororities. Put them together, we found that out of 47 sampling locations, 8 buildings account for half of all positive detections. And 3 buildings, which you can notice here, never had a sample which tested positive. There's many factors that can affect the concentration of SARS-CoV-2 in wastewater, like the number of COVID-19 cases in the buildings, the frequency and timing of shedding into the sewer, and the concentrations in the body entering in the wastewater, variations in flow, and the travel time of wastewater from buildings to the sampling point. The application of this surveillance can be greatly impacted temporary reflection of various laws in the wastewater. So a lot of factors we have to consider. Some papers have reported that there's uncertainty in this method, that sampling approaches knowledge on the person, there's a lack of knowledge on the persistence of SARS-CoV-2 in wastewater, and the low recovery efficiency of the various concentration method, and the molecular assays may be a problem. Also, the most important thing for us now is the sampling approaches. Why it is important? Because poorly designed strategies can introduce barriers in data interpretation, like the false negative results. So how can we choose the sampling approaches? The graph sampling or the composite sampling? The graph sampling is the method that we collected at one location at one point in time. This means samples not collected under under controlled conditions, so the data may be not reliable. And the composite sample is a sample which we combine the water together, and it is a time period. We can get the representative results from this sampling approach. There's one case that we feel weird at the beginning we start sampling, because there's one time that we know one building have the active cases, but we get negative results from the wastewater, because I think most of the time we choose to grab the samples, not using the composite sampling method. Also, this is the results. We use the composite sampling approach to do the samples, to taking the samples. This figure shows the concentration variation of SARS-CoV-2 in different time periods. From these three figures from one building, which you can see the concentration of the SARS-CoV-2 changed a lot. Also, you can notice that when we use the 2-hour composite sample and the 4-hour composite sample, the concentration of the SARS-CoV-2 is much smaller than the 1-hour composite sample. These three figures are from other three buildings. From this, the composite sampling, we can suggest that if you want to grab the sample, it is better to get the sample in the morning time. And in the future, I want to say what I have to do is to normalize the data and find one model to find the relationship between the active cases and gene copies of SARS-CoV-2 in the wastewater. And since now in our campus, most of students have been vaccinated, we want to know how does vaccine development affect the wastewater surveillance. Also, we have sent our samples to Maryland using NAVA 6000 to sequencing some of our samples. And also in our lab, we use the MiSeq to sequencing our samples. So we want to use the sequencing to track mutation associated with existing and new variants. Okay, thank you so much for taking time to listen for this, and I'm very happy to take time for some questions.
Thanks so much, Yi. That was a great presentation. I'd like to remind everyone, if you have any questions, please add them into the Q&A box. While people are thinking about those questions and adding them in, we can get started with a couple questions that I had. Let's see, so to go back to your methods briefly, you mentioned that you are concentrating the wastewater samples with both centrifugation and filtration. I was curious, are you processing, so if you can maybe just describe that in a little more detail, and most importantly, I was wondering, are you processing the solids portion of your sample or the liquid, and have you looked at both of those portions at all in your method development?
We are using the liquids, not the solids, because we get the method from the MIT, so they think at the beginning we think it is maybe there's much more virus in the water, but now we know that maybe it is better to get the samples from the solid, but there's much more inhibitors in the solids. So we think it is much easier for us to take the samples from the liquid. We have 14,700 samples per week to deal with, so we choose the liquid to get the results.
Great, thanks. All right, the next question is about sharing your data. And I'll add something onto this question as well. So first, how overall is the wastewater data shared with students at University of Tennessee?
No, we don't share the
Okay.
wastewater data with the students.
Not, yeah, okay, so it's all internal just with the university administration?
Yes.
Yeah, and I was wondering maybe a similar question to the one that Ali posed to Ayella. You know, you're obviously doing sampling. In some cases, it seems really localized at a single building level within the building itself. Have you, outside of communicating the data to the students, you know, have you received any either pushback or interesting responses from the students about the work that you're doing, or what has your sort of experience been with how people are perceiving this this testing at such a sort of a pretty localized level?
I think they are good with our works. They think we have made the contribution to the safety of the campus. So we at one time, there's one student may want to know our data, the results from our results, but I don't know, they can't get it. The university don't want us to publish it.
Sure. And so then to follow up on that again, there's another question and a similar, so if you're sharing this data just with the university administration, can you comment a little on what level of data you provide to them? Do you give them the quantitative information like you mentioned here with these stoplights, or do you give them positive, negative, you know, what level of detail do you provide for their response efforts?
We will report all the results, include the negative results and the positive results. We will give them the numbers of each building.
So the concentrations of the genes in each building, or just whether or not they came? Yes. Okay. Okay. Thanks.
So we actually,
this
is a combined project. So we are doing saliva testing, pooled saliva testing, so it does not have to be CLIA certified. So when we find stuff in a building, and I notify the director of the Student Health Center every day of our results, and the 2 or 3 vice chancellors, et cetera. But also the people that are doing the saliva testing. So we do pooled saliva testing, and they may be 4 or 5 of pooled samples. We're not identifying individual people, so that does not require CLIA certification. So from the pooled samples, pooled saliva tests, which is being done by a different group, we then actually, if we find any positive pools, we send all the students in that pool over to the Student Health Center to get tested. Okay. So that saved us a lot of time and a lot of money. Plus, we have one person when we're collecting all these samples that is dressed out in a Tyvek suit. Okay. So it's very obvious. And I think that has really helped us keep our levels very low after that initial spike in the fall. And in fact, we are getting so low now that we probably will end all the wastewater testing at the end of June, maybe sooner. Because we're practically down to, even this week, we've got 3 dorms that had trace amounts. And then all the sororities and fraternities are negative. So we're getting to the end game here. So, and we are also testing, we're using the Yale, if you didn't talk about
Yeah, I don't talk about
the fact that we're using 2 Yale primers so that we can test for both the UK and the Brazil strain. And we are finding the UK strain to some extent, but that's to be expected. We don't think we saw it in the fall, but this spring we are seeing it, so.
Okay, great. Thank you so much for that. And we have a similar, maybe a related question here about some of the data you presented. It looked like, and maybe this is from the subsets of data that were shown, I'm not totally sure, that most of your samples appear positive or at least weakly, sort of a low positive signal. When you're, yeah, so this, I think it's probably this plot here. Are these lowest bars, are those negatives or are those weak positives?
No, so those are negatives, but since our limit for detection is 20, okay, I automatically put everything down to 20 when it's negative, okay?
Okay.
Because I don't want to mess up our statistics on that.
Mm-hmm. Great. Okay, I think we've kind of gotten to all of our questions here. And we're a little.
And by the way, we have 2 presentations in the World Microbe Conference in June related to this in much more detail.
Awesome. Thanks so much. Yeah, these were really great presentations today. And I think since we're pretty much done with our questions, we can wrap things up. If you have any additional questions for Yi or Ayella, please feel free to, of course, reach out after the webinar. But with that, I want to say thank you again to both of our presenters and to everyone for tuning in this morning. We'll have our next webinar in 2 weeks. So thank you again and hope everyone has a great day. Thank you.
Thank you. Thank you.
So that was fine.